Hello, today we will demonstrate how does the koala genome impact the taste of eucalyptus. The koala is the last extant member of the
Phascolarctidae
family and can be found in the eastern and southern coastal regions of Australia. Koalas are arboreal folivores who also spend most of their time just below the canopy of eucalyptus trees upon which they feed. This presentation will explore the basic cell structure and biology of eucalyptus leaves, how and why koalas choose particular leaves, how these leaves are digested and the toxins metabolized, and finally, the key gene families that are responsible for these aforementioned abilities. The koala's diet consists solely of eucalyptus leaves, an evergreen species that is native to Australia and remains green and functional all throughout its growing seasons. The structure of the eucalyptus leaves include all commonly found cells such as the mesophyll and the stomata, encased in a lipid and hydrocarbon polymer layer called the cuticles. Chloroplasts are also abundant throughout the mesophyll, generating energy through photosynthesis and changing positions based on the environmental stresses. Additionally, eucalyptus leaves are commonly found to be covered in epicuticular wax, a film that surrounds the cuticles and is composed of both plant primary and secondary metabolites. The primary metabolites found on the surface are the free sugars, polyols, malic acid, and the GABA. Additionally, it is also comprised of the available nitrogen and the fiber-bound nitrogen, which is important for the nutritional needs of the koalas, as a herbivore diet naturally does not contain an abundant amount of all necessary substances such as nitrogen and protein. The amount of nitrogen available was thought to show an influence on the koala's dietary choices when it comes to choosing the leaves, even when it comes from the same species of eucalyptus. The eucalyptus plant secondary metabolites are toxic to koalas and are composed mainly of essential oils and long-chain ketones that are disposed of via their specialized metabolism. These PSMs, rather than the primary metabolites, are thought to be the reason why koalas select leaves and are also present within the epicuticular wax. This is believed to be true since research on other eucalyptus consumers such as certain insects select leaves based on their tendencies to perceive sources from longer distances, factoring in the smell of non-volatile substances such as wax. Koalas also tend to base their choices on taste, such as the level of bitterness perceived by the TAS2R gene family, which are activated by the eucalyptus PSMs. Meaning the more bitter the taste, the more PSMs are present and the more energy they have to expand to metabolize it. Which means, like us, it's a good indicator to avoid that food source.
Eucalyptus leaves have high fiber, low nutritional value. Also, eucalyptus leaves are rich in secondary metabolites, such as terpenoids, which are fatal to most mammals. But eucalyptus are still the main diet of koala. Why is that? Koala's digestive system is especially adapted to these eucalyptus leaves, which contains toxic chemicals. This is generally believed that these toxins are produced by eucalyptus in order to prevent leaf-eating animals from eating the leaves. And the poorer the eucalyptus grows, the more toxins are produced. This may be the reason why most koalas only eat a few eucalyptus leaves and try to avoid living in the certain eucalyptus forest. A koala eats about 200 to 500 grams of eucalyptus leaves every day. Koalas' teeth are also suitable for handling these special foods. The sharp, long incisors are responsible for clamping the eucalyptus leaves from the tree, while the molars are responsible for cutting and grinding. The gap between the front teeth and the molars allow the koala's tongue to efficiently stir and mix the food mass in the mouth. Koalas are very picky with their food. In Australia, there are more than 600 species of eucalyptus, but koalas are only interested in a few of them. In some areas, koalas even eat only 1 to 3 types of eucalyptus to maintain their lives. Of course, there are also some other types of leaves, including non-eucalyptus plants, which are occasionally eaten by koala in small amounts or used as a cushion or sleeping pads. Koala's intestines is specially for digesting eucalyptus fibers. Other animals such as humans also have intestines, but comparing with koala, which are 2 meters long, they are nothing like the same. Millions of microorganisms in the intestines break down the fiber in food into nutrients that koalas can absorb. Koalas rely on specialized gut microbes to break down the tannins and other toxic compounds. Otherwise, they will make eucalyptus leaves difficult to digest. The toxic eucalyptus essential oils are metabolized by phase 1 and phase 2 enzyme before reaching to the hindguts and absorbed. Baby koalas absorb their microorganisms from their mothers by eating a special type of feces called pap. Nevertheless, only 25% of the food eaten by koalas is digested and absorbed.
The koala's diet is highly selective, feeding solely on the foliage from a preferred collection of eucalyptus trees. In 2018, researchers Johnson RN et al. Isolated and sequenced the koala's genome, giving rise to a 3.19 gigabase pair model available on the NCBI database under the name far-seen UNSW v4.1. Interestingly, the koala has 2n equals 16 chromosomes, which differs from the postulated conserved 2n equals 14 karyotype suggested in Marsupial Cytogenetics 1989. Johnson reports genetic variation in various regions of the koala's genome when compared to other Australian mammals, marsupials and eutherians. They describe development in the koala's vomeronasal receptor type 1 gene group, which augments their ability in detecting non-volatile odorous substances. Koalas are known to smell leaves before consumption, suggesting that their olfactory chemoreceptor function aids in curation, perhaps by detecting plant secondary metabolites, including toxins and terpene and phenol structural groups. The ability for koalas to withstand a diet so concentrated with such toxins stems from cytochrome P450 gene superfamily expansion. CYP has been identified in several other domains of life, including but not limited to bacteria, fungi, and plants. As a result, these genes have been extensively studied, showing that CYP has a high isoenzyme multiplicity across many species. Genes from CYP are responsible for encoding major enzymes that facilitate the many biochemical pathways in toxin metabolism. These enzymes have the particular role of carrying out the aforementioned phase 1 in metabolizing the toxic compounds found in eucalyptus. One crucial gene family that reveals a lot about the taste of eucalyptus is the type 2 taste receptor genes. These genes, which are also found in humans, are characterized as chemosentry sentries, vital in protecting the body against toxins and metabolic and infectious threats. Researchers have identified that the koala has more TAS2R genes than any other Australian marsupial and most other mammals, and that the type 1 taste receptor genes were intact and functional. It is suggested that the behavioral response of V1R genes attempt to regulate the concentration of PSMs ingested. The expansion in the TAS2R family is able to detect bitter compounds in PSMs, but the existence of non-pseudogenized TAS1R genes points towards a portion of sweetness or umami in the eucalyptus.
We can conclude that the koala's digestive system is a factor of their choice of food. The special structure of their intestine and the special enzyme allow koalas to break down the toxic PSM from eucalyptus.
The selection of eucalyptus leaves are based primarily on olfactory detection with influences from genomic expression for taste, specifically to the plant's secondary metabolites. The degree of bitterness from the PSMs, such as the essential oils and the ketones, determines the avoidance of those leaves, whilst the primary metabolites, such as sugars, contribute to the overall sweetness of the eucalyptus leaves and thus the level of desirability.
The flavor of these PSMs is attributed to the gene receptors TAS1R and TAS2R that detect the sweetness and umami and bitterness.